Today, I am diving into a method for variant segregation in rare disease when we cannot get DNA samples from both parents. Imagine this family. A child has a rare variant in a dominant gene matching with their symptoms perfectly. To confirm this variant is making the child sick, so we can look for possible treatments, we need to confirm this variant de novo. Typically, this is done through sequencing both parents. However, we cannot get mother's DNA, so we cannot segregate this variant. To solve this, we have developed a method using adaptive sequencing. Here is how it works. We collect a group of trio families with short-read data. We nanopore sequence the children and pass their reads through our bioinformatics pipeline. For each phase block, we choose a variant as the potential mutation and assume one parent as available. We fit a linear regression model and make a prediction, which is compared with trio data. After doing this for all phase blocks, we calculate the statistics. We manually examine phase blocks to make sure when our prediction is incorrect, we can recognize them. When false discovery rate was controlled below 1%, we observed phasing errors that could be categorized into these 6 ways. They were all visually identifiable. Using this method, we could determine the parental haplotype of 70% variants. Over 99% of our prediction agree with trio. When we could not determine the haplotype, often it was because the phase block had a very few phase variants. Then for families needing segregation, we nanopore sequence the children and pass their reads through the same pipeline. This time, we directly apply our model to make a prediction. We also inspect the entire phase block. This family has a de novo variant in a dominant gene. When we assume mother is unavailable, proband's haplotype with the wild-type allele is almost identical to father's haplotype. The difference is caused by a technical artifact. This means proband's mutant allele is on the maternal haplotype. Because mother is assumed unavailable, we cannot confirm de novo. In the same family, when we assume mother is available, proband's haplotype with the mutant allele is almost identical to mother's haplotype. The difference is caused by the mutant allele itself. Because the mutant allele is on the maternal haplotype and absent in mother, we prove this variant de novo. Today, I have shown how we segregate variants when we cannot get DNA samples from both parents. An advantage of this approach is to identify the parental haplotype of de novo variants, which is unknown with trio data. A limitation of our study is our phasing tool not considering structural variants and DNA methylation. We want to do more testing. This includes making our pipeline faster and more automated. Massive thanks to everyone who's involved in this project. Thank you very much for listening.